Welcome viewers to my channel A to YouTube. It's me Dr. Afzal to you here for discussing topic on health education, research, etc. Today's topic to you here is on carbohydrate, digestion, absorption and clinical significance. It is one of the important topic of MBBS, MDMS or NEET preparation biochemistry exam point of view. So let's begin the topic. So whatever the carbohydrate diet we are taking, it contains either polysaccharide, disaccharide or monosaccharides. So the polysaccharide may be starch or glycogen. Disaccharides present in the food may be sucrose, maltose, isomaltose or lactose. Monosaccharide can be glucose, fructose, galactose and pentose. So these all are the constituents present in the carbohydrate rich diet, whatever we are taking as a raw food in our diet. Now, how digestion of carbohydrates occurs in our body? We have different enzymes. One of the important enzyme for digesting carbohydrate is amylase. The other one is disaccharidase. We have different types of amylase in our body, and the function of amylase is to break down polysaccharides into disaccharides, and for this we have salivary amylase, which is secreted from the parotid gland, and pancreatic amylase, which is secreted from the pancreas. Now coming to the disaccharides, this enzyme act on the disaccharides and break it into monosaccharides, and to break down this there are different types of disaccharides present in our body. So the examples are maltase, isomaltase, lactase and sucrase. So the name given according to the substrate on which these disaccharide acts. Now we see this is the diagrammatical representation of our digestive tract and how the carbohydrate digests in our body. So the digestion starts from the mouth and it goes into the stomach and the small intestine. So we have the three important sites for the digestion of carbohydrates, and the important question is where the digestion starts of carbohydrate. So the answer will be digestion of carbohydrate starts itself in the mouth. So when the food reaches the mouth, the digestion of carbohydrate starts by the secretion of the salivary amylase from this parotid gland. Here we are showing the salivary gland here, and food which comes in the mouth due to the reflex activity, the saliva is secreted into the mouth and this salivary amylase will act on the carbohydrate present in the food. Now then from the mouth the food will reach to the stomach, then from the stomach to the small intestine. Here from the mouth it will reach to the stomach and then to the small intestine. So this is the three important sites for the digestion of carbohydrate. Now we will see one by one. So as I told you, the digestion of carbohydrate starts in the mouth, and when due to the reflex activity, if carbohydrate is present in the food, then salivary amylase will be secreted as saliva, and during the mastication the salivary amylase which is present in the saliva will act on the carbohydrate, which is whatever it is present in the food. And the salivary amylase is also known as ptyalin because it is secreted from the parotid gland. Now what are the different characteristics of this salivary amylase? It is also known as alpha amylase. So this is secreted from the parotid gland, which is present in the buccal cavity or mouth, and it requires this alpha amylase or salivary amylase requires chloride ion for its activation, and the optimum pH for this enzyme alpha amylase to act on the carbohydrate is 6.7 and the range is 6.6 to 6.8. So optimum pH is required in our oral cavity to act this alpha amylase on the carbohydrate present in the food. So this alpha amylase under this activation of chloride ion and optimum pH act on the alpha 1,4 glycosidic linkage present in the polysaccharide molecules of the carbohydrate diet and hydrolyzes this bond. So it acts as a hydrolase enzyme. So it acts on this bond and break down the polysaccharides into the different constituent. So the action of this alpha amylase will stop when the food will reach from the oral cavity to the stomach. Why? Because of the pH. Because in the oral cavity, optimum pH which is required for its action is 6.7, whereas in the stomach, when the food reaches into the stomach, because of HCl present in the stomach, pH falls to 3. So the activity of amylase stops in the stomach. Now, what is digested in the stomach, whatever which we are taking as a carbohydrate diet? No enzyme in gastric juice is present to break the glycosidic bond. So it is one of the important clinching point here. We have in the stomach no specific enzyme present in the gastric mucosa to break any type of carbohydrate in the stomach. We have HCl in the stomach to break down or hydrolyze the sucrose. So HCl will act on the sucrose which is present in the carbohydrate diet, and it will break down into the monosaccharide, glucose and fructose. So what happens when the food will pass to the duodenum or small intestine from the stomach? Look at here, food reaches to the stomach. After action of the HCl, the food will go into the small intestine, first into the duodenum. So here the pancreatic juices will be secreted because of the reflex activity, and the pancreatic amylase will act on these food molecules. So pancreatic amylase, which is also known as amylopsin, which is similar to the salivary amylase, will act on in the duodenum itself, and so the carbohydrate breakdown will start efficiently in the duodenum. So the characteristic of pancreatic amylase is it is called as alpha amylase, and the optimum pH required is 7.1, which is slightly higher than the salivary amylase, because in the pancreas, in the small intestine, the alkaline pH is required. So because of this alkalinity, nature given pancreatic amylase a specific optimum pH of 7.1 to act on the carbohydrate. And like salivary amylase, it requires chloride ion for its activation. It is similar to the salivary amylase. Now it is also, the action is also similar to the salivary amylase. It will hydrolyze the alpha 1,4 glycosidic linkage of the polysaccharide molecules, and it will break down the polysaccharide molecule into the different monosaccharides or disaccharides. So in the note we have this similarity of pancreatic amylase with this salivary amylase. Both these amylase are isoenzyme, one of the important example of isoenzyme. So these two amylase, salivary as well as pancreatic amylase, are examples of isoenzymes. But the difference in these two isoenzymes is only in the optimum pH. The optimum pH of pancreatic amylase is slightly higher than the salivary amylase, which is pancreatic amylase optimum pH is 7.1, whereas for the salivary amylase it is slightly low, which is 6.8. And the similarity is both the enzyme requires chloride ion for its activation, and the site of action is also similar. Both this enzyme will hydrolyze alpha 1,4 glycosidic linkage. So it is again the important MCQ question for your exam, either in the MBBS prof or in the PG exam. The site of action of pancreatic amylase or salivary amylase. So the answer is alpha 1,4 glycosidic linkage, not on the 1,6 linkage. Now the action of pancreatic amylase. This pancreatic amylase, as I told, it will act on the polysaccharide So the example of polysaccharide is starch and glycogen, which is present in the carbohydrate food. So the pancreatic amylase will act on this and it will break down the polysaccharide into maltose, isomaltose or dextrine and different oligosaccharides. So digestion part of the carbohydrate mainly occur in the small intestine. So the main site of digestion of carbohydrate is the small intestine. Why? Because the main action of the pancreatic amylase, because the most of the digestion of the carbohydrate is done by the pancreatic amylase, because we are taking lots of polysaccharide present in the carbohydrate diet, and the complete digestion of carbohydrate occurs in the small intestine. Why? Because food stays for the longer time in the small intestine. That is why it is called as it is the major site of digestion of the carbohydrate in the small intestine. So what are disaccharides? So what are the different disaccharides which are secreted in the small intestine? So the disaccharides enzyme are secreted from the brush border epithelium of the intestinal mucosal cell. So in the intestinal mucosal cells we have the brush border villi cells, epithelial cells which is present in the mucosa cell of the GIT tract, which secrete these enzymes. There are 4 different types of major types of disaccharides. The important one is sucrase, maltase, isomaltase and lactase. So the action of disaccharides is maltase will act on the maltose and it will break maltose into glucose and 2 molecules of glucose. Isomaltase will act on the isomaltose and it will break into 2 molecules of glucose. Lactase will act on the lactose and it will break lactose into glucose and galactose. Sucrase will act on the sucrose and it will produce 1 molecule of glucose and fructose. So the end product will be monosaccharide, which can be easily absorbed from the GIT tracts. So after digestion, after the action of disaccharides, we have in the GIT glucose, fructose and galactose as a monosaccharide, which can be easily absorbed in the GIT tract. Now we will see the carbohydrate absorption, how our body absorbs carbohydrate, what are the different mechanisms which act on this monosaccharide to absorb in our body. So we have 3 mechanisms here in our body to absorb the carbohydrate. So monosaccharide after digestion is present in the small intestine, which can be absorbed by passive diffusion, facilitated diffusion, or which is also known as carrier mediated diffusion or active transport. So in the picture here, passive transport, example of passive transport is diffusion, which is passive diffusion as well as facilitated diffusion, but the example of active transport is this one. So what is the major difference is in the passive transport, in the passive diffusion, there is no carrier molecule is required and the molecule can be passed through the biological membrane according to this number of molecule present in the, so no osmotic difference or partial difference is required. But in the facilitated diffusion, the carrier molecule in the biological membrane is required to transport the molecules or carbohydrate molecule from outside into the inside, and it is slightly faster than the simple passive diffusion. Whereas in the active transport we require the carrier molecule as well as it requires energy to transport the molecule from outside to inside or from inside to outside. So here no pressure gradient or osmotic pressure is required to pass the molecule, so it can transport in either way. So this is some important differences between the 3 mechanisms of absorption of carbohydrate. So for the glucose transporter, we have specific glucose transporters in our body. So mainly it is of 2 types. sodium dependent transporter and sodium independent transporter. So the sodium dependent transporter is called as SGLT, which is named according to their name, sodium glucose transporter, and sodium independent transporter is also known as GLUT. So GLUT is for glucose transporter. So GLU came from glucose and T for transporter. Now what are the different glucose transporter in our body? So there are more than 14 different types of GLUT transporter, but the important one is these 6. So we will see here. So the GLUT1 is present in the blood-brain barrier and heart. So in the heart it is present in the very lesser extent. GLUT2 is present in the liver, pancreas and small intestine. GLUT3 is present in brain, neurons and sperm. GLUT4, which is very much important, this is the exceptional one, which is insulin dependent, whereas rest all are insulin independent. So GLUT4 is insulin dependent. So it is present in skeletal muscle, adipose tissue and heart. GLUT5, it is present in the small intestine and kidney, whereas GLUT7 transporter for the glucose is present only in liver. So to remember the site of all these glucose transporter, we have the mnemonics here. See, GLUT came from the came from glucose and T for transporter. So we have different 14 types of GLUT transporter. So the important one, we will remember the site. So GLUT1 is present in these 3. So for 1, everyone wants to be on the top. So the mnemonics is everyone want to be 1. So B stands for RBC and blood-brain barrier. So for GLUT1, we have to remember everyone want to be 1. So B stands for blood-brain barrier and blood. Like this, GLUT2, see the picture here, 2 kid which are here, pan is there, 2 pan is there, and both are in live position, in live of eating something. So 2 stand for GLUT2 and 2 kid here, kid for kidney, pan is for pancreas, in is for intestine and live is for liver. So kid pan in live, 2 kid pan in live is the mnemonic for the site of GLUT2 receptor. For GLUT3, see the 3 is written here, and 3 represent the structure of neuron or ganglion. So the neuron majorly present in the brain. So the site of GLUT3 is brain and neuron, and sperm you have to remember. Now the GLUT4, as I told, it is the exceptional one and the most important one, because GLUT4 is a insulin dependent. So we have to remember 4N. 4 is for GLUT4 and N for insulin dependent, and it is present in the muscle and adipose tissue. So F can be stand for fat also. Fat means adipose. So it is present in skeletal muscle, adipose tissue and heart, to the lesser extent in the heart. So this is the mnemonics to remember the different site of glucose transporter present in our body. Now what is the mechanism of glucose absorption in the muscle or adipose tissue? So this is the structure of myocyte or adipocyte cell. In the myocyte or adipocyte cell, we have the membrane, biliary membrane. So here insulin receptor is there, because for the glucose absorption we have GLUT4, which is the insulin dependent. So these 2 cells has GLUT4, which is insulin dependent. So these cells will have insulin receptor as well as insulin receptor in the cell membrane, whereas GLUT4 in the inside the cell. So whenever the glucose molecule will come, so glucose molecule is represented here. Whenever the glucose level will be high in our blood, so insulin will be activated and it will reach to the muscle or adipocyte cell. So it will bind to the receptor, alpha type of receptor, and it will activate. It will activate by phosphorylation reaction, and it will activate the PI3 kinase system, and through this GLUT4 receptor from inside will reach to the peripheral site, or it will reach to the cell membrane. So the GLUT4 came to the biological membrane or the membrane of the myocyte or adipocyte cell. So by the action of GLUT4, all the glucose molecule which is present outside will enter into the muscle cell or adipocyte cells. So this is the mechanism of glucose absorption in the skeletal muscle, adipose tissue, as well as in the heart cell through the GLUT4 receptor. Now we will see the mechanism of glucose absorption in intestine, how glucose is absorbed in the GIT tract. So we will draw the diagram here. This is the intestinal lumen, and here the capillary blood will flow, and this is the intestinal epithelial cell. And in the intestinal lumen, after the breakdown, after the action of the different enzyme, glucose or different monosaccharide will come for their absorption, and after glucose or monosaccharide reach the intestinal lumen, the receptor which is present in the intestinal epithelial cell will be activated, which is the sodium dependent glucose transporter, SGLT. As it is a secondary transporter, it will require energy, and it acts through sodium potassium ATPase channel. So when the glucose molecule reaches the intestinal epithelial cell, this SGLT will be activated, and because of this activation it will activate this sodium potassium ATPase channel also, and it will pull 3 sodium ion from inside the intestinal epithelium into the capillary blood, and to maintain this 3 sodium deficiency from the capillary blood, 2 potassium will pulled inside the intestinal or epithelial cells, and for this action it requires the ATP. That is why it is a active transport. So here 3 sodium is outside and 2 potassium inside. Still we have 1 positive ion deficiency. So to maintain this deficiency, our intestinal epithelial cell will require this deficiency to be filled out. So for this sodium ion along with glucose will come inside the intestinal epithelial cell to maintain the positive ion deficiency. So by this, glucose will enter inside the cell through sodium dependent glucose transporter. So here glucose entered into the intestinal epithelial cell, and sodium ion will maintain the positive ion deficiency. Somehow the glucose, and that is why it is called as secondary active transport, because this channel doesn't have any active transport. Here ATP is not required. It will activate the other channel, which is sodium potassium ATPase channel, for their action. That is why it is known as secondary active, not a primary active transporter. It is an example of secondary active transporter. Now from inside the intestinal epithelial cell, this glucose molecule will activate the GLUT2 receptor which is present in the intestinal epithelial cell, and this glucose molecule will come into the capillary blood. Now to the lesser extent, other than sodium dependent glucose transporter, some of the glucose molecule will enter through the GLUT2 receptor, and it will reaches to the capillary blood. So for the absorption of glucose in the GIT, in the intestine, we have mainly sodium glucose transporter, and very in lesser amount GLUT2 is required. So the major receptor or major transporter required for the absorption of glucose is sodium dependent glucose transporter, and GLUT2 is also required for lesser amount. And this GLUT2 is an example of facilitated transporter. So there are 2 mechanisms: secondary active mechanism, which is the example of which example is sodium glucose transporter as well as facilitated transporter. So both are required, active and passive, both process involved in the absorption of glucose in our GIT. Now we will see the clinical application associated with carbohydrate digestion and absorption. So what are the different diseases which are there, which hamper because of digestion and absorption? So the first disease which is associated with this topic is diarrhea or severe dehydration or severe vomiting, in which the dehydration occurs. For this we require oral rehydration therapy, and it is also known as ORS treatment. Why we gave name S here? O for oral, R for rehydration, and S for salt. Why we called it salt? Because in the treatment of dehydration, either in the diarrhea or in the severe vomiting or in the heat stroke, the electrolyte imbalance occurs, and because of electrolyte imbalance there are severe deficiency of different electrolytes, water as well as carbohydrate. So for the treatment, for the dehydration treatment, we require oral rehydration as a salt. We supplement, or as a oral rehydration we give salt. So this is the WHO recommendation amount. So we give the sodium chloride, dextrose or glucose, 10% glucose, potassium chloride. So this is the salt we give in calculated amount to the patient. What is the purpose of giving this salt? Because we saw in the mechanism of glucose absorption, we require this sodium ion as well as glucose for their absorption. So in severe diarrhea or vomiting, there is lack of sodium ion, there is decrease in the water, there is decrease in the potassium ion. So because of this deficiency, this channel will be hampered, impaired, so there will be decrease in the glucose absorption. That is why the drowsiness, weakness occurs in the patient of diarrhea vomiting. So we give this oral rehydration salt for the treatment of diarrhea vomiting. So this is the important clinical application in this topic. Now the other disease which is associated for the carbohydrate digestion absorption is lactose intolerance. As the name suggests, it is the intolerance to the lactose. It is of two types. It may be congenital as well as acquired. So congenital may be from the birth. So in the congenital lactose intolerance, there will be a complete absence of or deficiency of lactase enzyme. So the lactase is required to break down the lactose present in the diet into glucose and galactose. So if this enzyme is not there, there will be increase in the lactose in our body. So there will be development of intolerance to the lactose. So in the infant or in the early infancy, this disease is very much common, specifically in some child where this congenital lactase deficiency is there. So it can be diagnosed in the early infancy as well. And milk feeding, whenever we provide the milk, as you can see that the child cries because of the symptoms produced because of increase in the lactose in their body. So what are the different symptoms as present in the lactose intolerance? So there will be the abdominal cramp or abdominal pain, distension, diarrhea, constipation or increased flatulence. So why distension occurs? So when the lactose is accumulated in the intestine, it is acted upon by the bacteria which is present in our body in the intestine or GIT tract. So it will act on those lactose and it starts increasing the gas. So there will be increase in the carbon dioxide and hydrogen, which causes the distension and flatulence. And because of this, there will be increase in lactose 2 carbon and 3 carbon compound in the GIT tract, and these compounds are very much osmotically active. so which will accumulate, which will withdraw the water from the intestinal mucosa, and it will lead to the osmotic diarrhea. So how we will diagnose this lactose intolerance? We have the 4 different methods. So the qualitative test is Benedict test in the urine. So from the urine we can diagnose the lactose intolerance by doing the Benedict test. We will see the color reaction in the urine. If it turns green, yellow, orange or red, we can diagnose the lactose present in the urine because of lactose intolerance. We can do the stool acidity test, hydrogen breath test and mucosal biopsy. It is an invasive procedure to diagnose the lactose intolerance, and it is specific to detect the enzyme lactase deficiency in the cell. Now how we will manage the lactose intolerance? So we can manage by avoiding the dairy product. We will not give the dairy product which contains high concentration of lactose. So we will give only those dairy product which has very less amount of lactose, or lactose free milk which is present, which is now commercially available. We can give the soya milk to the infant or to the adult who is suffering from lactose intolerance. So this is how we can manage the lactose intolerance. That's all for today. Thank you for watching my channel A2 YouTube. If you like this video, please don't forget to like button below this video, and also subscribe to my channel to get my new videos here on health education, research, etc. Till then,